Hello, everyone. Welcome back to part 2 of our lecture on energy. So on last class, we talked a little bit about energy and what types of energy exist. We established the laws of thermodynamics, and we also identified the units of energy. So what exactly is the energy currency of the cell? Let us move forward. The energy currency of the cell is ATP. In all living cells, energy is packed into ATP molecules, okay? And the energy stored in ATP is available for immediate use, okay? So when we consume foods, the goal that we're trying to do with cellular respiration is break down glucose, metabolize glucose in order to release the energy that's trapped in those chemical bonds, okay? So ATP is a temporary storage place for energy that is readily available to do work. All right. There are other long-term storage molecules in our body, such as lipids and so forth, but ATP is that storage molecule for which those energy is available to do work. So let's talk a little bit about ATP. You should be familiar with the structure of ATP. It is a nucleotide consisting of 3 main parts. It's got that 5-carbon sugar, ribose. It's got 3 phosphate groups, and it's got a nitrogenous base, adenine. So if you look at this structure, it looks very similar to the other nucleotides that we've talked about when we talked about the structure of DNA. In this case, we've got 3 phosphate groups here. We're going to look to see how this ATP molecule is created from its ADP precursor. All right, so you should be familiar with the structure of ATP. And this term, this acronym ATP stands for adenosine triphosphate. Okay, that triphosphate is referring to the presence of those 3 phosphate molecules. Okay. So ATP donates energy. So the energy in ATP is donated through the transfer of a phosphate group. So what happens is a process known as phosphorylation. This is a reaction in which a phosphate group is transferred to another compound. So in the process of the creation of this ATP molecule, we start with ADP, adenosine diphosphate, which indicates that we have 2 phosphate groups present. There's going to be another phosphate group added here, and this is how we get the creation of our ATP. So there's this ADP ATP cycle that happens. Okay, so this energy from our foods, when we're breaking those chemical bonds and metabolizing glucose, there are processes going on for which this ATP is created. And we understand again that the energy that's stored in these ATP molecules is energy that is available to do cellular work, right? We established that energy is required to carry out all of the biological processes that take place within the cell. So another factor in this process of creating energy, or not creating energy, because we don't create energy, but releasing energy that's stored, is the presence of enzymes, okay? Enzymes are a method for cells to regulate the rate of chemical reactions. So that's what we've been talking about in this unit. We've been talking about energy and metabolism. We said that metabolism is the general term for all of the chemical reactions that takes place within an organism. Well, enzymes are a way for us to regulate and speed up these reactions, all right? So we refer to enzymes as biological catalysts. All right. And in this case, these biological catalysts are going to function to increase the rate or the speed of a chemical reaction. So we're thinking about this. These chemical reactions that are taking place within living things, they're taking place for a reason, in some response to the needs of the cell. So we want this process to happen quickly and efficiently. Enzymes allow us to speed those reactions up. Okay. And so cells, we understand that the cells require a constant release or supply of energy, right? So we must regulate the release of energy in order to meet, we need to regulate the release of energy in order to meet the energy requirements for metabolic activity, right? So enzymes are a key control point for responding to the energy needs of the cell. So let us take a look at how enzymes actually work. So we refer to them again as biological catalysts. All right. And they function by lowering the amount of energy that is required for this reaction to take place. The energy that's required for a reaction to take place is the activation energy, right? And so we say that all reactions have an energy barrier, right? This is the amount of energy that is required in order for this chemical reaction to take place. That is the activation energy. All reactions are going to have a baseline requirement of energy in order for that reaction to take place. And so what happens with an enzyme being present in this reaction is that it's going to therefore lower or reduce the amount of energy that's actually necessary for this reaction to take place, thereby allowing this reaction to take place a lot faster. So enzymes function effectively by lowering the amount of activation energy required for a reaction. Okay, so enzymes speed up chemical reactions by lowering the activation energy necessary to initiate the chemical reaction. So we need to understand two things. That one, the presence of enzymes do not remove the need for energy. Okay, and so the second point is that energy must be available for this reaction to start. Okay, so this reaction must be able to begin even in the absence of the enzyme. But what this enzyme is doing is coming in and lowering the amount of energy that's actually going to be required in order for this reaction to take place. Okay, so understand mechanistically that enzymes function by lowering the amount of activation energy that is necessary to initiate a chemical reaction. So if we look at this diagram here, we have two reactions, okay? We have one taking place here on the left panel, and then we have another one over here on the right. The reaction that's taking place over here on the left is referred to as an uncatalyzed reaction because there is no enzyme present in this reaction, okay? Over here on the right, though, we do have an enzyme present, so this is an enzyme catalyzed reaction. All right, so you should be able to understand and read and interpret what's happening in these two chemical reactions, okay? This one is uncatalyzed because there is no enzyme present. This one is catalyzed. So let's look and see what's happening here. On the y-axis, we have the total amount of energy that is being utilized. On the x-axis, we have the reaction pathway taking place. And we know that a chemical reaction takes place. We have a reactant on one side and we generate products on the other side after the chemical reaction has occurred. In order for us to go from reactant to product, there is some activation energy that is required. And so here in this uncatalyzed reaction, we have a normal amount of activation energy that's required, okay? This graphing right here represents how much energy will be necessary to complete this reaction, okay? Going from reactant to product. This is an uncatalyzed reaction. This is the normal amount of energy that is required. However, if we take a look over here for this same reaction, right? This chemical reaction wants to go from reactant to product. What we see here is that there is a reduced or lower amount of activation energy here that's been lowered by the presence of this enzyme. Okay, so this is our enzyme catalyzed reaction. So we should be able to look here and compare and see that the presence of this enzyme here in this reaction has effectively lowered the amount of activation energy that's required for this reaction to take place and our product to be generated. Take a moment and make sure you understand what has happened here. Okay, so how do enzymes actually do their jobs? Enzyme must form what we call an enzyme substrate complex, right? So we know that enzymes are proteins, okay? Enzymes fluence reactions by forming a complex with the substrate. The substrate is the substance for which this enzyme acts upon. So this enzyme substrate complex must be formed in order for this enzyme to become active, okay? And so every enzyme has what we call an active site. This is the region on the enzyme where it binds to the substrate. So think about this enzyme as a lock. Every lock has a very specific key that's going to open this lock. This lock has very specific grooves and cavities for the key that has been cut for this lock. This is the same case for our enzyme and its respective substrate. Your enzyme has a place where this substrate binds, and that place is called its active site, or some textbooks refer to it as a catalytic site. The active site is the place on the protein, on this enzyme molecule, where the substrate binds, okay? And it's going to form an enzyme substrate complex. Once this enzyme substrate complex forms, this enzyme can become active now and function as a catalyst in this reaction. So here we have a schematic that shows us an enzyme, okay, this protein structure right here that has very specific conformations, these grooves and cavities that are very specific for a substrate. The substrates bind the enzyme and form what we call an enzyme substrate complex, okay? This enzyme substrate complex renders this enzyme active, and the reaction can occur and the product will be created. After the catalyst has functioned, the enzyme is able to drop off and become available to catalyze another reaction. So we want to understand that enzymes have specificity. The shape of the active site on the enzyme closely resembles the shape of the substrate, okay? And that's because most enzymes are highly specific to the substrates that they require. Okay, so not every substrate will be able to fit every enzyme. Okay, so we're understanding that most enzymes are only going to catalyze a few reactions or one particular reaction. Therefore, there has to be a very specific substrate available to bind this enzyme to catalyze that reaction. Okay, so we have our enzyme substrate complex formed when we have the specific substrate for this reaction, and then our products can be generated. And so then there are some other factors that can influence enzymatic activity. Okay, enzymes can have cofactors. All right, so some enzymes contain only that protein portion. The protein portion alone is referred to as the apoenzyme. Okay, The apoenzyme alone is usually not enough to get the full catalytic activity of that enzyme. All right, so some enzymes have additional components known as a cofactor. Okay. In this case, neither the apoenzyme or the cofactor has catalytic activity alone. It is only when they are combined does this enzyme become functional. So we are bearing in mind that there are multiple factors that can influence the overall activity of the enzyme. Okay. And so structurally, again, we're understanding that the enzyme is a protein. The protein portion alone, without its bound substrate, is going to be referred to as the apoenzyme. And then we're understanding that enzymes can have additional barriers that's required here in terms of cofactors and so forth in order to become functional. So when we're thinking about enzyme activity, we need to understand that there are other predetermined conditions that must be met in order for this enzyme to function optimally. Okay, so optimal conditions for enzymes are necessary if we're going to get the optimal catalytic activity of this enzyme. All right, so we understand that in chemical reactions, enzymes work best under certain defined conditions. Okay, these are what we refer to as optimal conditions. Optimal conditions exist when under the optimal conditions, this is when we get the rate of reaction at its fastest. Okay, so long as we meet these predetermined parameters for this enzyme, we're going to get this rate of reaction at its peak. Okay, so some of the defined conditions can include temperature, pH, and ion concentration. So these are all factors that we can manipulate and control in a chemical reaction in order to create the most optimal environment for this enzyme. Okay, so we're going to stop right here and think about some of the things that we've learned. Take a moment to go back and understand this ATP molecule and its structure as the energy currency of the cell. Energy is going to be stored in ATP for immediate use. Okay. And then we go on and understand that this ATP is fueling chemical reactions. And so another piece of this puzzle is that reactions can be influenced by the presence of enzymes. Enzymes and metabolism go hand in hand. So it is important that you understand what enzymes are and how they function. We introduced to you the concept of sorry about that. We got disconnected for a moment, but I was wanting to just kind of recap what we had talked about in terms of enzymes and how they function, what their structures are. We're understanding that enzymes require a substrate in order to become active. When the enzyme binds with this substrate, it forms an enzyme substrate complex, and then it can become catalytically active. And then we just also highlighted that there are predetermined conditions in order for this enzyme to function at its best. Okay, there are many factors that can influence enzyme activity, such as temperature. Enzymes are very temperature sensitive. These reactions can be pH sensitive, ion concentration. These are all factors that can influence enzyme activity. We're going to stop here and continue on next class.